Hello, everyone. Thanks for joining. Today, I'll be talking about advancing multimodal neuromechanics and understanding of brain dynamics of human locomotion. I'm an assistant professor in the Mechanical and Aerospace Engineering Department, and I'm also part of the Bionics and Adapt clusters. I direct the Biomechanics, Rehabilitation, and Interdisciplinary Neuroscience lab, and this is a picture of my awesome lab space. So I started at UCF about 4.5 years ago, and key achievements so far are an NSF Career Award, NIHR1, and I'm also an alumni of the National Academy of Engineering Frontiers of Engineering program. My research focuses on neuromechanics, the interplay of neuroscience and biomechanics, human locomotion, in particular walking and balance control, locomotor adaptation, which is the process of adapting walking patterns, and our approach is to use a mobile brain and body imaging method. And we hope that this will help inform gait rehabilitation and healthy aging. I currently teach biomedical engineering courses in our growing BME program. And in my undergraduate biomechanics class, I tried to use lecture demonstrations that my graduate students helped me with, and also engage students in different UCF STEM activities. So the motivation for my research is related to understanding the human brain and mobility. So first, there's a need to uncover the underlying brain processes and sensory motor control principles of human locomotion and adaptation, and to develop new sensors and methodologies to study the brain in action. So in my lab, we use a dual layer electroencephalography, EEG approach. So what we've done is we've mechanically coupled two EEG electrodes such that one electrode interfaces with the scalp and measures the traditional EEG signals, which are electrical activities on the scalp. And then the other outer-facing electrode interfaces with a conductive layer, and that allows us to measure an isolated, quote-unquote, ground truth motion artifact and noise. We've assembled a 128 dual-sided EEG system that we will be using when we can resume activities in the lab. The other motivation is related to mobility, which is strongly linked to quality of life. And being mobile means walking about in changing terrains and unexpected conditions, such as going for a hike in the mountains or taking my dog for her nightly walk. I'm interested in the preservation of mobility with aging, the restoration of mobility after injury or disease, and also augmenting locomotor capacity. My NIHR1 is on adaptation of brain and body responses to perturbations during gait in young and older adults. I have co-investigated at UC Davis and also here at UCF in nursing and statistics. And some specific questions include, what are the underlying brain processes crucial for being able to adapt to perturbations during human locomotion? And how do these brain processes conserve across locomotor tasks, like walking, recumbent stepping, and cycling? And how do they conserve with aging? So in this video, is an older adult responding to perturbations, which are it's just a brief increase in resistance indicated by the red arrow as he steps on a robotic stepper. And then every so often, there's a stride where there is no perturbation. We call those catch trials right there. And that allows us to probe whether subjects are forming an internal model and learning to anticipate for the perturbation. So we see in our data so far that young adults have distinct responses to the perturb steps and the catch steps. And this suggests that they are forming an internal model and that they're learning to anticipate for the perturbation. On the other hand, our older adults show a different strategy, and perhaps they are just stiffening their limbs and co-contracting their muscles to reject any perturbation. And when we look at the brain activity, we identify clusters in the anterior cingulate cortex, which is related to error detection and monitoring, and then also the left and right supplementary motor area, which we don't see in older adults. So we're trying to figure out what that means. Of course, we also apply perturbations when people are walking. We have a programmable split belt instrumented treadmill that can pitch up and down in real time and also translate side to side. This is a video of a split perturbation. So this is where the right belt speeds up briefly during right mid-stance, indicated by the green arrows. So they're very small And every so often we don't perturb them, which allows us to probe anticipatory controls. So using the equipment like motion capture system and ground reaction forces, we can examine biomechanical mechanisms of balance and walking controls. And in here we see that for the perturbations, initially the perturbations were destabilizing, so it created greater instability. And then over time, they learned to reduce their margin of stability and gain stability. And they did this primarily through how they controlled their center of mass versus where they're placing their feet. So this analysis is still ongoing. The other main project is my NSF Career Award on leveraging electroencephalography, EEG artifacts for multimodal neuromechanics. So typically, we use EEG to look at brain activity. So the goal is to eliminate EEG artifacts, which are the non-brain signals, to obtain high fidelity brain signals. So this video will show what EEG signals look like with different actions. So when the subject closes his eyes, there are these visible 10 hertz waveforms. Those are called alpha waves. And then when he clenches his jaw, there's these really large signals. And when he jerks his head, there's again a large signal that's created. And these other actions mask the underlying brain signals, the alpha waves. And that's what makes recording EEG during a dynamic task like walking challenging. So these artifacts are readily recorded. So rather than just getting rid of them, I propose that rather we could, instead of just discarding the artifacts, we could extract useful information from them based on knowledge of their sources. So we could use dual layer EEG to get the ground truth motion artifacts, and then using independent components analysis, ICA, we can separate the EEG signals into source signals for the eye, muscle, and the heart. And then combining that with machine learning, I hope we can estimate gait symmetry, eye gaze fixation, like where people are looking at, and then also metabolic cost, which is just the energy expended during movement. So I think EEG has the potential to predict biomechanics, but the converse is unlikely. Okay, so the vision for the future of neuromechanics, in my opinion, is, well, first let's consider what current multimodal neuromechanics looks like. So you have all these systems if you're lucky enough, and if you use them, it's time consuming. So you're talking about 3 to 5 hours for one data collection. And it's expensive if you're going to own all these systems and then sync them together and manage and process all the data. And it's really just kind of impractical. Like, I don't know anybody who has actually done an experiment with all the sensors and equipment that I've listed here. So now imagine in the future, we could just use dual layer EEG and machine learning, and we could get measures of the brain dynamics, gait symmetry, eye gaze fixation, and metabolic cost, so that we don't need to own or use all these systems. And I think this is possible. Again, it's part of my career award, but this effort would be greatly accelerated by collaborating with others with more experience in machine learning and deep learning. So transdisciplinary research needs team science, and that's new collaborations. I think this comes up a lot in the funding solicitations. So some target programs for me include Brain Initiative 2.0, specifically related to brain in action, non-invasive technologies, and new analytic approaches for complex and large data sets. At NSF, there are 2 programs that I'm targeting: Mind Machine Motor Nexus, which involves closing the loop. So in my case, perhaps brain-machine interfaces. And then there's also the SENSE program, which is on multimodal sensor systems for precision health enabled by data harnessing, artificial intelligence, and learning. My near-term projects right now are to prototype and evaluate different form factors of dual-sided EEG and EMG electrodes. So EMG is just electromyography, which measures the electrical signals on your muscles. This is aim 1 of my career award. And here I'd like to mention I'm also working with StretchMed Incorporated, which makes epidermal electrodes. I have students working on developing interactive art devices for lower limb neural rehabilitation. So far we have the PATE board, which is shown here, a jaw cycle, and an unnamed phone app in the works. And the idea is to investigate self-exploratory based rehabilitation that engages creative processes. Long-term projects include working with clinical populations, such as those with stroke or incomplete spinal cord injury. And then again, brain-machine interfaces, where perhaps we can try to tune perturbations and use different tasks to engage and enhance specific brain processes that might be beneficial for walking. Two main research or teaching goals are to add EEG modules to biomedical engineering courses and to strengthen an infrastructure of advocates for girls in STEM by having members of the girls support network share STEM experiences with STEM, and by creating a sense of community among those advocates in a program I'm calling Neuromechanics Girls. So here's a picture of my lab members. We call ourselves the Brain Dept, and thanks for listening.
Very good.